So it's my pleasure to introduce Dr. Francis Collins, who will be joining us just by recorded remarks for this workshop. He is the director of the U.S. National Institutes of Health. He is the former director of the National Human Genome Research Institute, and he was also the leader of the Human Genome Project. So we owe a lot to him, and so please give your attention to him.
Good morning. I'm Dr. Francis Collins, director of the National Institutes of Health, the NIH, and we are excited to be a co-sponsor of this important workshop, Learning Meaningful Representations of Life. I wish I could be there with you today in Vancouver for this effort to forge new connections and deepen existing ties between the biomedical research and artificial intelligence communities. But unfortunately today I need to be at NIH's headquarters in Bethesda, Maryland for a meeting of the advisory committee to the director. That's a big deal. In fact, it was about this time last year that this distinguished committee announced formation of a working group on, guess what, artificial intelligence. The NIH, which happens to be the world's leading public supporter of biomedical research, is already investing pretty heavily in AI, but we need to do more. And do more smarter. So I've asked this new working group to help us develop an overarching vision for this area that holds such enormous promise for scientific discovery and improving human health. Not only will this group of forward-thinking experts chart a new vision, they will recommend strategies on how to go about achieving that vision. So over the past year, the AI working group has been hard at work fulfilling its charge, which includes identifying innovative cross-NIH opportunities in AI with broad reach and positive effects across many diverse fields, diseases, and research communities. Helping NIH build bridges between the computer science and biomedical research communities to establish ongoing interactions that move both fields forward. Facilitating the development of training and career pipelines that encourage computer scientists to work in biomedical research environments, as well biomedical folks to take greater advantage of computer science training opportunities. And last but not least, suggesting ways to address ethical considerations related to the use of AI in health-related research and care. Today's interdisciplinary workshop is just one of many innovative tools that the AI working group is using to carry out its mission. Hopefully, what goes on here will catalyze the exchange of creative ideas between biomedical researchers and computer scientists, relationships that we hope will grow and flourish with time. NIH is grateful to the organizers of this highly influential conference in Vancouver for recognizing the importance of our efforts and agreeing to include this workshop in their lineup. And I think they'll be glad they did. Looking over today's agenda, I see you'll be hearing from some of the best minds in both biology and in machine learning. Their presentations will give you a sneak peek of what the world might expect from the next generation of data-driven biological problem solving. Discussions will emphasize interpretable learning of structure and principles, and those themes will be applied to work on the level of the genome, molecule, cells, and phenotypes. I hope your experiences here today spark long-lasting collaborations that foster deep, productive interactions across the biomedical and computer science fields. I also encourage you to welcome participation from experts in the behavioral and social sciences, ethics, and many other disciplines essential for applying AI in ways that will turn scientific discovery into better health for all. In closing, I'd like to thank each of you for making the time to be part of this historic melding of communities. I look forward to reviewing the session readouts and to participating in more activities like this one in the future. So, without my causing any further obstruction, let's get this workshop started.
It is on. We'd also like to recognize Google Ventures, or GV, sorry, for their funding and helping to make sure that this workshop can actually happen. And we've asked Dr. Krishna Yashwant, a managing partner at GV and a practicing physician at Brigham and Women's Hospital, to give a few remarks as well.
All right. I'm a little jealous that I didn't also produce a YouTube video, but you get me in person here. Welcome. We're thrilled to see everyone who made it here today. Thank you for the introduction. My name is Krishna Yashwant. I'm a primary care doctor at Brigham and Women's Hospital in Boston. I'm a recovering computer scientist and a managing partner at Google Ventures, where I oversee our healthcare life sciences investment team. I've been looking forward to this workshop for several months. I've always found the most exciting things tend to happen at interfaces, and I think this room is full of interfaces between machine learning, biology, clinical medicine, academia, and entrepreneurship. So thank you all for coming out for this. I'd like to thank everyone who came together to plan such an amazing day for us. We have a truly world-class group of leaders and thinkers here in the room today, and I'm looking forward to a lot of thought-provoking discussion. In order to bring the best and brightest from around the world, GV sponsored travel grants that brought 137 researchers from 75 institutions across 21 countries. That's nearly 1 out of 4 people in this room today. Some specific people I want to call out for their energy and commitment to make all this happen include Ann Carpenter, Deb Marks, Elizabeth Wood, John Bloom, Ray Jones, and Yakir Reshef. I just want to take a moment for everybody to look around this room and just appreciate how much effort, thought, and time in the day that I know these people actually usually don't have, but they made the time to pull something together of this caliber. So can everybody join me for a moment and give this group a round of applause? If you see any of these folks around, buy them a drink as well. I also want to thank the many co-organizers, the discussion facilitators and speakers for making this happen, and of course, all those who traveled to present the posters and build community spanning machine learning and biology. Before we kick off with today's program, I want to remind you why we're here. We seem to be moving to a world where there's a lot more data about people, where perhaps nearly every person on the planet will be sequenced. It's not clear whether that's going to happen as a reaction to people getting sick and us wanting to diagnose their precise disease, or whether it'll be preemptive via perinatal screening, screening people before they're even born, or whether it'll be to decode our family heritage, or how it's happening, but that seems to be the direction we're moving. But what if beyond that, beyond sequencing every person, what if we started to think about what makes up the organism of a human, and think about all the different types of cells in each of our bodies? How many different types of cells are there even? What makes one different from another? How many different states can these cells exist in? How do we store this data? How do we organize it? How do we make sure that people have access to it? And how do we make sense of it? And how ultimately do we use these insights to prevent and cure diseases? I certainly don't know the answers to these questions, and I'm sure no one here fully does, although I'm expecting Aviv might have a lot of them. But perhaps together we can explore the interfaces between us to continue making headway in these important areas. When we start with an interdisciplinary approach and put different people and technologies together, we can begin to make significant progress in the way we care for patients and treat diseases. In every corner, we see the need for this intersection to continue to develop, and in my work at GV, I get to see what this looks like at the other end as we're getting ready to bring these sorts of insights and technologies to patients. But for any of that to be possible, we need to start with the sort of work we have before us today. Machine learning is going to help us understand who we are and translate that knowledge into making significant health advances possible. And I continue to be really excited about the direction we're headed. I'll admit I'm an optimist, and I admit there's a lot of hype around this dream, but from where I sit, we're at an incredibly exciting moment in time. The rate of progress is tremendous, and it will only continue to accelerate, thanks to everyone in this room here today. So with that, let's get started. I'll turn it back over to you, and let's get going.
Okay, right. Thank you very much. Thank you, Francis, out there. Okay, let's just move on, and a short announcement and a slight change in the keynote, which is Daphne's not going to be in conversation with Deborah Marks, but in fact with Barbara Engelhardt. And the 3 keynotes this morning are Aviv Regev, Max Welling and Daphne, who have all, in their own ways, forged whole fields and more. And I don't think really there's much to say. I think everybody in this room knows Aviv. Aviv is first up. I'm really not going to describe it. Aviv Regev from the Broad and from MIT. Welcome.
Well, I'll just stand here. I think it's safer. Or I'll move my laptop. I think it would be better if I moved it. We can do it. This is a little clunky, but it will work. I don't feel like standing, like going back and forth. So I do not actually know if everyone in this room knows me. I very much doubt that, but I do know a lot of people here, and it's a great moment in time to come. I was reflecting with David Hausler. Each of us was saying, what's the last time we actually came to NeurIPS? And mine was in 2010. Actually, Christina Leslie invited me to the workshop that's happening tomorrow in 2010. And one of the thoughts I had when I came and I gave my talk was that if we ever really want to do some of these problems in biology, we'd better have data that looks very different than what we have right now. And at least for me, my group and I and many people in my field have spent, I think, a lot of time at trying to get the right types of approaches so that we can do this kind of inference. And this is really going to be a major theme in my talk today. So I was asked by the organizers to highlight some of the ways that we might think about biology. That was a pretty broad description, and I'm going to try and do something about that. And so I'm going to describe biology maybe as the integration of multiple levels of organization. The fundamental unit of life is the cell. You cannot reduce below it, but the cells are made of molecules which are either encoded by genes or they're made by gene products using other components from the environment. The cells then interact with each other, and in multicellular organisms they make tissues that make organs, which all of them together account for the organism's physiology. The organisms make populations and ecologies, and on that evolution acts. And it's actually important to remember that this is not a stack, it's a circle, because evolution leads to selection on heritable variation, which are the genes themselves. And so biology seeks to understand this. That's basically what the field is about. It wants to understand life. And there are three kinds of questions that biologists ask themselves. The first is what, which is really a structural question at any of these levels. What is a biological entity actually made of? The second set of questions are how, and this includes both genetics, which is the basis for causality in biology, as well as mechanism, which is how something is actually implemented. And the final and very difficult question in biology is the why, which is a unique question in science and is unique to biology. You know, you can ask a functional question at any level of the ones that I've described. You can ask what does it do for the cell, and what does it do for the tissue, and what does it do for organism physiology. But in the end, it only makes sense through the lens of selection and evolution. Now, to answer the structure question, we need to not only map each of these layers, or each of the entities on each of those layers on their own, but we also need to map between them. And these mappings are, first of all, between different types of variables. You could have something that looks like an image and something that is actually a molecule that you have to map to each other. But they're also not linear, and they combine in all sorts of weird ways. As a result of that, for a very long time in actual biological practice, it was not really possible to infer this mapping. You had to actually measure it. Now, to answer the causal mechanism question, we need to understand how genes and gene products encode information that provides for these many other different layers. But again, just like with the mapping between the layers themselves, the genes can combine in many different ways. And these ways might be non-additive, and that's what biologists call genetic interactions. And again, until recently, we couldn't really predict what would happen when we would combine two genes together or manipulate two genes together. We had to actually measure them. And then finally, to answer the function question, ultimately we need to understand evolution, the trace of what has happened and the potential of what might happen next under certain selective pressures. But the fodder for evolution is huge, it's vast. And our ability to experiment on it is, minuscule, not just in terms of the scale of the problem, but also in terms of time scales. Evolution might happen over billions of years, and we're kind of, we don't have that time scale in our, in our lifetimes. And so in theory, and I think this goes to Krishna's point, the space of possibilities is really enormous in biology, right? Except that most of these possibilities are never realized. And you can make many kinds of these calculations. This is just one set of examples. The problem is that we don't know up front which ones would matter and which ones do not. And that at least until recently, we thought that we would have to actually measure everything in order to see it. And so one of the biggest questions for right now is how can we solve problems like these systematically, even if we cannot do exhaustive experiments that test every possible option. And in fact, I want to make the case, again, already I think foreshadowed by Krishna, that this bigness that seemed to feel like a huge problem for biology in the past, all of a sudden seems like its biggest virtue in how we can find our way out of this problem. But it's useful to think about what people have done until now. And so one option, which biologists have often taken, and very wisely so, is to actually use some information that they already had in order to restrict the search space. So instead of testing, say, all possible mutated sequences, just probe around the ones that we already know do something in a biological system. Instead of exploring all hypothetical ways in which genes can combine to a program, look at particular subsets of genes that you already know do something together. Instead of testing all possible interactions, which you actually can't do experimentally, instead of testing all possible interactions over, sets of 5 genes out of the 20,000, choose those that you already think are doing something interesting. And instead of just looking at some sequences, design assays that look at particular one. And this idea was used again and again and again and again in biology. These have all been great approaches. They pretty much gave us everything we know so far. So we should treat them with much respect, but it's not clear that they would give us a general answer to this problem. And so today, I'm going to talk about how we can think about new experimental designs that are inspired by algorithms and that in turn provide algorithms or allow algorithms to represent and understand biology better. And I'm going to kick it off by giving an intuitive idea of how an experiment in biology might look to someone who is not an experimental biologist at all. So imagine that biological discovery is about figuring out which painting I'm hiding in this frame. Every experiment is going to be about removing one tile, and I can only remove some tiles and far fewer tiles than the total number of tiles that are hiding the picture. Now, I know something about this picture. This picture is very structured. It's going to have patches of color, and it also has tons of background, and I'm not really interested in this background. It's going to be some, grayish, brownish type of color. And so until recently, all we could do is really remove a very small number of tiles. So if we already knew that there was some color in some corner because we had some biological knowledge, here's red, because I know there are patches, I should dig around this red. If I know there's yellow here, then I should dig around the yellow because I know, again, that it exists. If I took the same number of tiles and just randomly thrown them at my picture, I wouldn't be able to tell anything, and most of the time I would just hit background. That's traditional experimental biology. However, what if I could uncover a lot more tiles? Not nearly as many as the whole picture, but just a much larger number of them. Well, one possibility is to actually throw them at my board completely randomly. And if you are fond of modern art, you would be able to tell by this point that this is likely a painting by Miro. And if you're really fond, you would know that it is actually in the Broad Museum in Los Angeles. I chose that, of course, on purpose. So these are the kinds of experiments that you might think about if you weren't necessarily a biologist. But if you were a machine learning person, all of this explanation is kind of, yeah, that's obvious. That's what we do all the time, right? We fill in those blanks. Who needs to measure everything in life? But that's not how we thought about experimental biology until recently. And so doing this in biology is not typical, but it's a great opportunity, and from which we've already been reaping benefits. And today I'm going to describe it in 3 parts: cells, mechanisms, and convolutions, and all of them are going to be just very quick and short vignettes. The first part is going to be cells. The first case study is single-cell RNA-Seq. I'm sure most of you have heard about it, if only because you went to Dana Peer's keynote a couple of days ago. And you might have known that it's been a fantastic advance for our field, and from which we learned a lot of biological insight. But for experimental biologists, this was actually a very uncomfortable exercise. This is because one of the main experimental decisions that we made early on, which led to things like drop-seq and massively parallel single-cell RNA-seq more generally, was to favor having very sparse and noisy data from a very large number of cells. But the natural thing to do in biology at that time would have been to have very deep and accurate data from a very small number of cells. And the reason to favor the large number of cells with sparse and noisy data was mathematical or statistical. We knew that there was a lot of structure in how genes co-vary with each other within and between cells. And so we should be able, right, to fill in analytically everything and see the patterns, even if the data itself is noisy. And we cannot do that if we have a small number of cells. And so this was my first example of experimental design made for inference, and it really paid off big time, because from such data we now know that we can learn remarkably many different representations, and many of them are on these posters around from my quick, perusal in the morning. And I'm going to take you through some of these very briefly and very generally, and highlight actually those places where we struggle. And so the first one is that we can uncover discrete types of cells, from very common ones to exquisitely rare ones that we didn't even know existed before we have the data in hand. And we've done that in systems like the retina and the airway epithelium and the mucosa, or the lining of the colon. But cell type is one of those concepts. I guess the poster is taking exception to some of the things I'm saying. But cell types is one of those concepts that is intuitively really compelling, but is actually really difficult to nail down in biology. And one of the hopes is that, of course, the data would drive you there. However, you get into these circular situations. Yes, you found some entity based on data. How do you kind of know that it is real? What does validation mean is one of the biggest questions that we're going to grapple with. Some of it is, it should be reproducible. A second aspect should be, it should be, complete. If I looked at more data, I would kind of come back with the same kinds of answer. But another aspect of it, I would probably want to see it in some other representation based on some other data besides the original one, because they should all line up with each other, because in the end, there's only one cell with its RNA, its proteins, its structures, and everything in it. We can also represent how cells change over time through differentiation from a single fertilized egg as they respond to their environment. Here, the cell's identity is really based on what are its possible fates, what might still happen to it, but also where might it have come from, what is its possible history or histories. And there are examples here for the zebrafish embryo, for reprogramming in iPS cells, and again for the colon epithelium. Again, there are challenges. For example, each one cell at any given moment in time is actually undergoing multiple simultaneous dynamical changes that reflect different kinds of things. It's differentiating, it's responding to this thing in the environment and to that thing in the environment. Different cells might be undergoing the same type of changes. And also the possible fate of the cell is not always that easily mapped to the actual realized lineage, meaning the actual series of cell division events that led to that cell. And again, we need ways to confirm these inferences. It's lovely to have them. It doesn't actually mean they're real. And our models have now started to far outstrip our experimental validation capabilities. We can also define the identity of the cells based on where they are, where they're located anatomically, meaning in which organ, where they are in the finer histology or structures of the tissues, and even in the direct cellular neighbors that they have. All of these things can be recovered from their profiles. But again, tackling how those scales, the gross one to the finest one, actually relate to each other is not always obvious. As you go from one individual to another and you take a specimen, how do you know that you actually took it from the same place? For this, you need a common coordinate framework. And we don't actually have it, and we especially don't have it to creatures like us that actually don't all look exactly the same. And so we actually need to learn those coordinate frameworks too. The location of the cell is dynamic. Some cells move, but moreover, the cells themselves and everything that surrounds them changes all the time. And finally, we need to find repeatable patterns. Because when we find repeatable patterns, be it a community of cells and the structure that they're embedded in, they often help us identify units of actual tissue function. And then finally for the cells, even though cells are the key unit that I highlighted, many behaviors don't obey the boundaries of the cell. There is much variation within a cell type and often spanning a spectrum. And so these patterns can be shared also across subsets of cells, as we see here for certain types of innate lymphoid cells. These are immune cells. They all have an internal pattern with them, and it repeats between them. And they're better represented actually at the level of gene programs rather than at the level of cell types or cell categories. But again, there are challenges here too. How do we disentangle these multiple programs, some of which might interact with each other? Is a cell merely, you know, is the cell type, the first thing I started with, merely just one kind of program, maybe a very prominent one, but by no means the only thing you can say about a cell? To what extent are these spectra biology, meaning they're real and they reflect something meaningful, and to what extent are they measurement noise? And how do we interpret the basis of the variation when it's neither space or time? Okay, so the last thing I want you to remember about cells is that all of these identities and these facets that I highlighted exist in a single cell at once. But most of our methods and models, including many that I think we will see on these posters, try to focus on one of them at once, which makes it difficult to combine it together. But one cell has a type, a location, a history, and possible fates. It's also undergoing these multiple transitions all at once. It's also located at these different resolutions all at once, and these different aspects interact with each other. And this means that the fundamental challenge for us is not just disentanglement, but also understanding these relationships and, in the end, interpreting all of these dimensions. And I'm going to highlight here just one example. These are cells from the small intestine on the left, and they carry programs that highlight their point in differentiation, but they also carry programs that highlight where they're located in the small intestine. It's separated to 3 different anatomical sections, and these 2 things interact with each other because differentiation is different a little bit depending on where you actually are. So with all these caveats in mind, this first exercise in design for inference really led us to the sense that we should be able to complete an atlas or a reference map of all cell identities, which we often allude to as like a periodic table of the cells. It's a lovely metaphor. We make beautiful graphics, but the periodic table actually had an underlying theory, and as a result of that, it was predictive. Can we hope for something like that in biology? Not just describing everything we already measured, but predicting, for example, things we've never seen. And so this leads me to the second part, which focuses on mechanism, where a big challenge has been the many different ways in which genes can combine to control cells. There's many other challenges. I'm going to focus on this one, and I'm just going to use this relatively simplified view where we just think about levels of RNA expressed from the genomes, those things we measured in the single cells. We have upstream molecular circuits that control downstream gene expression, and they do this through regulatory sequences that affect RNA production. And we want to discover these kinds of circuits. But one big edge we have in biology that we have not looked at thus far by looking at the individual cells is that we have the ability to intervene in the causes, which we can do by perturbing one or more causal entity at a time. This is called genetics. We can do it naturally, by just using natural genetic variation, courtesy of evolution, or we can do it in an engineered way in the lab by introducing the perturbations ourselves. Now, then we can do a measurement of the effect, for example, measure gene expression. The problem, as I noted, is that there are way too many options to do these perturbations compared to what we would ever be able to do experimentally, and I want to emphasize that there are not enough cells in the world. Okay? It's not that we can become better and smarter and cheaper at experiments. There aren't enough cells. So over the last several years, we started developing some additional designs for inference. And again, by quick perusal of the posters, I saw some like-minded spirits. And I'm going to give you a very few brief vignettes. I'm going to start with how regulatory sequences control gene expression. One way to go about it is look at a lot of examples: regulatory sequence, associated expression, learn a model. People have been trying to do that for years, but they've been limited by the kind of data that they had in hand. Either they used what evolution giveth, sequences that are already there, or they synthesize sequences, usually starting from a known starting point of a regulatory sequence, and they were limited in how many of those they could sequence, maybe tens of thousands, maybe 100,000. That would be considered a very big experiment. Neither of this is enough. So here our solution was to make a very simple assay, measure the noisy expression level of hundreds of millions of sequence examples, simply by making random sequences. Making random sequences is super easy. It's really easy to do, but it makes biologists very uncomfortable, because random sequence, why would it even work? Okay? You can put it into cells, and you would have to see how much expression they drive. And I think up front, people thought they would drive no expression. Turns out they drive any level of expression you can imagine, from very low to extremely high. The question is, can you learn biology from it? One way to learn is to build a very interpretable model, one that's very decipherable and is explicit and defined in biochemical terms. You can put in a lot of biological details, because even though the model has, hundreds of thousands of parameters, which is a lot for models like these, the problem we have data, in the hundreds of millions of sequences. And this kind of model gives us very detailed mechanisms. It highlights very precise ways in which regulatory proteins interact with the DNA and give us expression levels. And some of these things are things that have not been properly appreciated before. For example, the model showed, and we confirmed this with experiments, that weak interactions that we usually completely ignore by models are actually predominant in the way that regulation happens. Now we can ask, how does it do prediction? Well, it does a pretty good job. It generalizes to other random sequences, about 92%. It even does a pretty good job on real yeast sequences, which are anything but random, and it does about 85%. But of course, if what you want to do prediction, and I'm giving a talk here, then you should probably change your modeling approach. So we did. What if we learned a model that is focused on prediction? But it's still inspired by biological principles. So it should be able to have the right characteristics so that it captures motif specificity and interactions, transcription factor scanning, long-range interactions. For the biologist here, all of these things matter a great deal. Activity after the binding and so on. So you try to find the right modeling paradigms that actually reflect these kinds of biological behaviors. And you train a model, by now with 3-point-something million parameters, and it reduces your error by 33%, 98% of for random sequence, 92% accuracy for native sequences. But the most critical thing is not that it gives you nice predictions. That's good. It shows the model is doing something. But that it actually gives you real distinct value. If you think like an engineer, you can use a model that's trained like this to search for sequences that would have specific expression levels that you want them to have. For example, very high and very low expression, and we did it. And when we synthesize and measure them, we actually managed to go beyond the extremes of what native sequences could do. But even more interesting for us, in terms of understanding biology, this model carries unique value for us, because it allows us, if expression of a gene is a proxy for something in evolution like fitness, then it allows us to actually describe a full fitness landscape on which we can ask very fundamental evolutionary questions. And that was not possible before. Okay, second and even more rapid vignette, consider expression programs. The idea here is very simple. Don't measure the expression of each gene individually. Measure composites of genes, or the sums of gene expression, and then recover the individual measurements with compressed sensing. As a mathematical idea and implementation, this existed in the world. But in biology, again, this is uncomfortable for people. What did we spend the last 20 years of genomics doing? Measuring every variable. And it was valuable and critical for us. So the main assumption here is that gene expression is structured and that the expression profiles of any given cell or sample can be described by a combination of a small number of modules. So first we showed theoretically with simulations on real gene expression data that this should work. Compressed measurements can be used to accurately cluster profiles. We can decompress them to get the correct abundances, including blindly, and we can even use them to phenotype perturbations. But we wanted to show that this is not just a parlor trick, it's actually useful for something. And one of the places where these things become the most useful is when you can't measure every variable. You're limited in the number of variables that you can measure, which is often the case in situ measurements in the tissue. So we used mixtures of probes to measure gene composites, and then we decompressed, and because we're decompressing images, we actually did it in a latent space. This is what composite measurements look like. That if you're a neuroscientist, you know that the motor cortex doesn't look like that for any gene. But these are the decompressed images. They were never measured individually, these pictures that I'm showing you. They were measured only through decompression. They are seen only through decompression, and they fit very well both the previously measured images in the Allen Brain Atlas for these, as well as direct validations that we've done when we individually measured the same genes on the same section at the end of the process. My third vignette is going to be about using genetics to identify causes. To assay gene functions, especially when related to gene regulation, we use today an assay that we call Perturb-Seq, which we developed a few years ago. And in these assays, we deliver interventions, genetic interventions into cells, for example, with CRISPR. We barcode them as RNA so we can recover them later. And at the end of the experiment, we profile every cell. So we know which intervention we put into that cell and what was the outcome in terms of gene expression. And this is what a result like this might look like. This is out of a simple linear model with some basic regularization. And what is important about this image is how structured it is. Okay? So the target genes, which are in the rows, are all co-regulated in these neat programs that respond in the same way across the perturbations. And also the genes that we perturbed, which are in the column, they impact those genes in very similar ways, again, making these modular structures. So now we can use this structure in order to make more efficient experiments by no longer measuring any individual gene or individual perturbation, but measuring their compositions. And there's all sorts of ways that you can imagine doing this, either by profiling little groups of cells or having more perturbations within one cell. I'm going to show you just one concept of overloading cells and measuring them as sums instead of measuring them individually, and compare them side by side with a traditional experiment where every cell with its own perturbation was measured individually, and this is in an inflammatory response. So we measure them the two ways. We take the compressed data and we use compressed factorization, which was very recently published, to learn which modules of co-functional perturbations regulate which modules of the co-regulated genes. The first sanity check is that the decompressed expression is 97% correlated between the traditional and the compressed experiment. And then when we look at the impact of the 5 known major positive and negative regulators in this system on 4 major known targets, those are also consistent. And then we extend that to the top 15 effects. And then we extend it and we go further down to the top 56. And even though the signal gradually is degrading, nevertheless, it actually works. And then very briefly, for my last 2 minutes, I'm going to say a few words about convolutions. I'm only using it metaphorically, but I do think that the metaphor actually helps here. And this is about how the layers connect together, not mechanistically, but really phenotypically, in how the phenotype of one layer relates to the phenotype of another layer and gives us an integrated phenotypic view. And I'm going to return for this to our beloved cells. And in my illustrative example, I want to ask, can we define the cells not just based on their intrinsic properties? Gene expression is an intrinsic property. The shape of the cells, anything about the molecules inside it is an intrinsic property. But cells also have extrinsic properties because they sit inside a tissue and they have neighbors and there's structure to the tissue around them. Can we bring both of these pieces together in order to define the cells for us? The example I'm using is from melanoma. We label the tissue section here from tumors with the expression of multiple markers that highlight specific cell types, including cancer cells and immune cells. And then we learn a model by learning a new kind of embedding for us, which combines the gene expression of the individual cells and an image around them, of everything around them, not segmented or anything like that beyond that. We learn an embedding for each of them individually, and then we learn a joint embedding, and for the joint embedding, we ask to predict the gene expression of the cells. Whereas for the embedding of the image alone, we just ask it to predict the image back, a standard autoencoder. There's some few minor bells and whistles involved. What is interesting is what we do with the embedding that we get. We call it in situ VEC, and we use it to cluster our cells, for example, and we can find distinctions that we wouldn't have recovered from the intrinsic expression alone. So, for example, a certain class of T cells that is very important in tumors are CD8 T cells. They are useful to attack the tumor cells and kill them, but unfortunately sometimes they don't get there. Turns out that the in situ VEC embedding took one group of them and split it into 3. And these 3 clusters make spatial and molecular sense. One cluster are those T cells that are actually directly proximal to the malignant cells that they're trying to engage with. And the second extreme is a cluster of T cells that make their way into what we call the cold niche, the part of the tumor that immune cells somehow cannot infiltrate. But these special T cells can, and we can see them, and we can only see them by considering their environment. And these patterns are persistent and they're generalized, even though these are tumors which are very different. Even within one person, they're different from each other if you have multiple tumors, and surely between different individuals, including individuals that the model has never seen in the training. This idea that we can combine molecular and structural features, or different kinds of molecular features, or different kinds of structural features, or multiples of them, is very native as a machine learning problem, and it will be extremely useful as a biology tool. It would save us a lot of experiments for sure, but it would also be a very fundamental fusion within biology. In biology, you often have to choose. Am I thinking like a tissue biologist or a molecular biologist or a cell biologist, even though in the actual biology, it's all just one thing. There isn't some, mysterious ether there. there are a lot of nonlinear transformations. But those are things that, this community knows what to do about. And so with this, I'm going to finish. I'm going to thank the people who did the work. I had the acknowledgement throughout the slides, and I'm not going to go through all of them because there are a lot, but I do want to highlight Carl de Boer and Ishit Vaishnav for the work on the sequences, Brian Cleary for all the work on the compression, and Ishit again for the work on the tissue, as well as a very large number of people in our group for the many, many studies on single cells. And I especially want to thank my partner in crime in the last several years, Orit Rosen, who is a biologist, and Anya Hupalovska, with whom I work on those graphics. Thank you very much.
Okay. Thanks, Aviv. That was completely fabulous. Could Max Welling make his way to the front, please? And we're not going to do questions because we have so many speakers today, but you can exploit the speakers and the people who work with the speakers at coffee break and going around the posters. Thanks so much, Aviv. Okay, here we have Max, who's just going to set up. If the posters fall down during the session, it adds to the liveliness. So perhaps we wait to coffee to put them back up so there's less noise. And we've got more sticky stuff and things at the front here. We want to come up in the coffee break. While Max is setting up, I just want to say that he actually inspired, I started my lab up, and his work inspired a whole new direction. My lab, so we're very grateful for his very articulate way of describing wonderful work. Okay, Max, take it away. Can you tell people where you're from? And
oh, sure. So my name is Max Welling. I'm in Amsterdam, work at the, I run the machine learning lab in Amsterdam, and I also working at Qualcomm. Yeah, it's a great honor for me to be here. It's been fantastic to see the uptake of variational autoencoders and in this community. And today I want to talk about sort of new developments in this field, and which I think are also going to be very useful for the biological sciences. And they're somewhat related to VAEs. I'll introduce VAEs again. I'll, discuss the new methods in light of the VAE. So let me start with a much more general sort of discussion about machine learning. So machine learning is just about, combining inductive biases with data, right? And the more inductive bias is, the more modeling assumptions you're putting in your model, the more, mistakes you're going to make, because you're going to, in some sense, be reliant on your own imagination, and all models are wrong. But it's a good thing to do if you don't have a huge amount of data. On the other end, in modern machine learning, for many applications, there is huge amounts of data. And in that case, you should be more relying on that data, because it's just hard to write down models that are correct in all little details. And generative modeling is in one side, in the inductive side part of the world. There's lots of inductive biases that you can inject. And you can do great out-of-domain generalization. I think this community, and in generally sciences, are mostly rooted in that side. But the modern machine learning view is that if you have a lot of data, you should sort of do deep learning, and I'll talk a little bit more about that. It's typically quite limited, because you have to, you can do well on a very limited domain, but then on that domain, and you can collect a lot of data on that domain, you can do incredibly well. So some of the the interesting things happen on the intersection of these two extremes. Now, in machine learning, we often forget that 99% of all the other sciences are doing something different than we are doing, which is they write down simulators where they encode their inductive biases of the world, or they write down graphical models or probabilistic programs, or ordinary and partial differential equations. They're huge fields that write down everything in differential equations. And also in machine learning, we used to do this like only 10 years ago or 5 years ago with graphical models. Most people forgot about graphical models, but that's the time that I grew up. And I think they're still extremely useful to use. So this is the other paradigm, the machine learning paradigm, which is, it's sort of the inverse of a generative model, where in a generative model you start with factors, latent factors, things that you can interpret and that are sort of at a semantic level, and you write down how that will generate data. Machine learning works in the opposite. You take your raw data, an image or a speech signal, and you push it through some black box neural network with millions or billions of parameters, and you predict those properties directly. So you say, this is a galaxy of this size, whatever, right? So it's the opposite of what we typically do in the sciences. So how are these two paradigms related? Well, they're related by very simple equations that you should all know, which is Bayes' rule. So in Bayes' rule, you have your generative model here, which says, given a class or given some latent variables that describe the world, how do I generate my data? So that's the data generative process. And then there's typically some priors over those as well. And that is proportional to the probability of those properties or the classes given the raw input. So here is your machine learning deep neural network, and here is your PDE or simulator or whatever. So the VAE was an idea actually that combined these two in a somewhat principled way. So in a VAE, on this side, you have your generative model. So here you start with a prior over latent variables, P, Z, and another prior over classes, let's say. And then you push it through a neural net to predict some parameters of a distribution, let's say a mean and a variance. And that will give you a latent variable of pixels. Basically, how do I generate a pixel X? And that would constitute an image, let's say here. And then the encoder is the opposite, which is you can think of as the discriminative model or the machine learning model, which is you take your raw image, your raw data, and you push it through another neural network to predict yet another set of parameters, means and variances, from which you then sample those latent factors, those interpretable semantic factors or classes, right? And here you can predict the classes, let's say, from those interpretable factors. And the cool thing is that these two things, they need each other to learn. So this, in order to learn this one, you need to know what Z is. And in order to produce Z, you use your encoder here to produce distributions over Z. If you have that distribution, you can sort of complete all the variables that you don't know, and you can train this just as a regression problem. So you can also view this guy here, your generative model, as a regularizer to train this discriminative model. If this is what you're interested in, then maybe it's a good regularizer to say that that should be the inverse of a generative model that I know. Because here is where you can store your knowledge, right? In this generative process is where you can put your knowledge. And so you can say, well, I'm interested in this mapping, but let me make sure that that at least approximates the inverse of a generative model that I know. Now in equations, if you want to translate that in equations, so the VAE basically looks at a joint distribution over Z and X. X is the, it's kind of interesting process here, everything comes down. So there's a joint distribution over Z and X, which is latent and observed. Here's my encoder, here's my data distribution. And there's a prior over Z, and this is my generative model. And I'm trying to get these two distributions to be the same under a KL measure. And I can decompose that into two parts. One is a KL between the data distribution and the model distribution, and a KL between, an expected KL between these posteriors. And so really to get it right, you need the distribution on X over the images to be the same, and you need these posteriors to be the same, right? So that's what a VAE tries to do in equations. And instead of having a neural network, you can stick your simulator there, right? And that's what you can all build these simulators, and you can put it there. Okay, so to sort of sum that up, there's white boxes, which are the interpretable models, the generative models. They're data efficient because you can inject your knowledge, say laws of physics. They're interpretable, all the variables mean something, and they generalize better out of domain. So because, for instance, you can have causal structure in them and stuff. The machine learning models are the opposite. They're more flexible. They're less biased to human imagination. right? We cannot get that right, so we need the data has the information, and you'd better to learn 1 billion parameters if you have the data to learn that. They're fast because you don't use Bayes' rule, and they're accurate because they predict precisely what you're also going to test on. This is typically what you're going to use your model for, and you don't want to first do, you know, an approximate sort of Bayes' rule in order to get to that. And maybe it's summed up by this quote, which I really like personally. It says, the ingredient, which is causality, should allow computer systems to choreograph a parsimonious and modular representation of their environment, interrogate that representation, distort it through active imagination, and finally answer what-if kind of questions. So this basically says that, you know, I can imagine a pink elephant biking on the moon, right? And I will never see that ever in my life. Nobody will see that. Yet, I can imagine that. And that's the power of a generative model. It's completely out of distribution, yet I can imagine it. And this is what Pearl, you know, why he sort of thinks that causality is so important. Okay, so now I'm going to switch gear a little bit. I'm going to now introduce something new, which is also presented here at NeurIPS. It's related, and I think it's going to be very useful for this community, and that's why I'm also very excited about it. And it's also related to VAEs in some sense. So the idea is that of inverse modeling, and you know, inverse modeling is actually very well known in the sciences, where you have some kind of forward model which says I have some object that I'm interested in but I can't measure, and then I have some forward generative model or an encoder, yes, that will then produce, say, a noisy measurement for me. Oh, it could also be the decoder, actually. Okay, so it produced a noisy measurement for me. And then but that's my generative process. This is the one that I know. Actually, that should be the decoder. I think I messed that up. And then the reverse, sort of going from the measurement back to the actual quantity of interest, is the inverse, and that's what you're interested in, right? You're trying to invert your generative model. That's what the VAE also did. Okay, there's plenty of examples for this. So here's one example in astronomy. You have radio images of the sky. You don't know them. But you can measure them, and so you have an array of antennas. They measure these frequencies, and then since you only have a finite set of these antennas, you have a very sort of sparse subsample of your Fourier measurements, and then from those Fourier measurements, you need to get back to that sort of image here. Now, if you would do just a naive inverse Fourier transform, you would get that's very bad. So you would need to clean it up with machine learning. And here's a really cool example with gravitational lensing. So you have a black hole which is in front of a galaxy, and it distorts the view of that galaxy, and you need to infer back that galaxy and the parameters of the black hole. So this was, this method was applied actually to that problem. And here's one maybe that's more important for health, which is MRI reconstruction. So you take, again, you're interested in this latent image of a brain that you don't know, but you're interested in, you're doing these measurements, which are, again, Fourier measurements, but it's very expensive, and you'll be in a machine for very, very long if you do all these measurements. So you do a small subset of them, and then if you do the naive inverse, you get a not so nice reconstruction, and then you use your deep learning tools to clean it up, to make it better. And there's here an example, an error correcting decoding. So you take some image and you add some additional parity check bits, you send them over a noisy channel, you receive this on the other end, and you use your inverse model to clean the whole thing up, and then you get the clean image out. So the traditional solution to problems like that are you write down your generative model. So here's your generative model. This describes how the data Y is generated from the latent object X. This is your observation model. You have some prior over what images of the world look like, right? So that's where your modeling assumptions go in, where, so basically your inductive biases sit here, and then you're going to write down some optimizer over X. You're going to try, okay, let's maximize this object to get the maximum likely X that still explains my data Y. And you can do gradient descent. And that's the, the simplest zeroth order sort of inverse modeling that you can do. You can also, do some more sophisticated, algorithms to try to find that maximum, like belief propagation or message passing. But typically, all of these are iterative optimization scheme, right? At every time step, you reconstruct the object of interest, and then you look at it, you evaluate it, and then you do it again and again and again until you get something really good. So the deep learning solution to this, which is again the opposite, right, in this duality that I described, the deep learning solution is collect a gazillion data points, X and Y, where X is the object that you're interested in a training set. So in a training set, you could imagine, you know, measuring everything there is to measure, and it'd be expensive, but you can do it. And your sort of observations, which could be the Fourier components that you measure, a small subset of the Fourier components. So you create tons and tons of these data, and then you train your favorite fat, big neural net, let's say a UNet of some kind, right, that, you know, does a whole lot of computation on a billion parameters to reconstruct for you, you know, the object X that you're interested in. Now, that works fine, but there's one thing that's blatantly obvious that's inefficient about this, which is that it doesn't use something that it knows, which is the forward model. It knows precisely how to go in the opposite direction, which is from X to Y. And this is that's only expressed through the data set. But why not actually stick it into this model directly and let it enjoy it while it's trying to reconstruct, you know, and do a better job? So that's the plan. And so if you've been sleeping or checking your email, maybe this is the one slide, you know, that you can take home. So wake up, which is what we're going to do is we're going to iterate that iterative reconstruction process, they're going from Y to X. We're going to view that as an RNN, a recurrent neural net, where every timestamp is basically one of these reconstruction steps. And now, so that's just a classical solution, right? It's just unrolling the algorithm that we already had. But now we're going to train a neural net based on data to correct the mistakes made by that classical solution. Because the classical solution was bound by our imagination, right? We can't write down really, really complex models. It's too the world is too complex. But it's in the data. So now we're going to ask the data to correct the mistakes that are made by the engineer solution, right, or the model solution. And the second ingredient, which is really important, is that in this process, we're going to embed the forward model, because we know what it is, and we should be using it. So basically, here is how that looks like. Here's your input data, Y, which are your Fourier measurements. I have some reconstruction step, and that reconstruction step should use this forward model. This is my generative model, Py of X. So it should use it in some way. And that's basically what we call the engineer's update. So that's basically, you know, the gradient descent on your model. And then we have a bunch of memory states, because we have that flexibility. We can store some information in some memory states. That's typical for an RNN. And then we have our neural net that's going to compute corrections to these engineer's updates. So instead of doing the actual engineer's update, we add a term which is learned from the data. So we wrap around a classical solution, we wrap a neural network solution. And then, of course, we can backpropagate through the whole construction. How am I doing on time?
Fine.
I'm fine still?
You're fine. You're fine.
Fine, yes. Okay, good. I heard something going off, so I feel good. Okay, so what's the relationship between a VAE and this new recurrent inference machine? So a VAE, it starts with an X that I observe. Then I have an encoder, QZ given X, that produces for me this latent object Z that I'm interested in. And I also have to learn a prior P of Z, because when I want to generate data, I need to have something to generate for. So I'm going to also fit some P of Z here. And then I have a P of X given Z, which is my decoder. And then I'm going to check how good it was, what I generated. I'm going to check it against the real data X. And then I have a loss function, and then I, descend on that loss function, right? Okay, so how does it look like for this new beast that I just described? Now, I should say, this is the version where typically it's a supervised problem. You have the pairs, and you can just do ordinary sort of regression. So this is a version where I don't have the supervised pair. So I don't have Z, I only have X. So it's sort of an improvement over VAE, if you want. And I should disclaim that I thought of this in the airplane, and you shouldn't take it too seriously, because we didn't try it. Okay, so the X so you start with your x, and then this path here is exactly the VAE path. So you go from x to z, from z to back to x, and you check your error, right? So that's exactly this process here. You check your error, and then you're going to hand this error back to the next stage. You're going to say, okay, you did really bad on these z's, right, that produced your x's. So these z's here are, really responsible for your mistake you're making here. And you're going to give that information to the next stage, and then it can use that to correct it. So now you're embedding your forward model in, in this process. You iterate this for a couple of times, and then at the end you spit out your final x, right? And then at the same time you have to train this prior, which is sitting here, and we do this in the usual elbow way. Have a KL between those, and you can train that. Okay, so that's maybe the way that these two are related. So let me then discuss the some experiments that we did at a high level. So here is a nonlinear chaotic dynamical system called a Lorentz attractor. So it's very complicated. So here you see basically so here's the trajectories of how, if you run that as a ODE. But it's chaotic because you can never predict whether you go left or right in this. And then we add noise to it, and then the task is to reconstruct the original lines here. So if you do what's called a neural net solution, which in this case was a graph neural net, you get this. So you don't have enough data to really get it right. And also the dynamics is just too complicated for this. Then the extended Kalman smoother, which is the sort of engineer solution, is a bit better in this case, but not perfect. And then if you combine the two in this model that we developed here, which you get a really, really good solution. So this is work by Victor Garcia and co-authors. So of course we also applied this to MRI image reconstruction. So this I already showed before, so let me skip it. Okay, so this looks very complicated, and it is somewhat complicated, but it is basically the implementation in some detail of what I just described at a high level. So you have some RNN which tries to reconstruct X, which is the image, and some memory states, and you have some, some update function which depends on the memory states and depends on the state X. And it depends on this forward model, which is this here. So it uses this forward model as information as it is iterating through this thing. And these functions are implemented as convolutions and GRUs and all these sort of things that we have in deep learning. And then you back propagate through the whole thing to get to train all these parameters. So it looks like this. If you write it down as a circuit, I don't really expect you to understand this, but it looks impressive, so we'll just show it. So here is the data. So we used 3 different MRI data sets, another brain with a different magnetic strength, and a knee, data set with knees and brains. And then here's what the data look like. So this is in Fourier space, so that doesn't look like anything, but it's like you have Fourier components, more sampled in the middle, which are the low frequency components, but they're subsampled at a level 4, 6, 8, 10 times. So which is really nice, because if you subsample at a level at 10 times, you can reconstruct at 10 times the speed. So you're in that machine only 1/10 of the time, which is, I think, good if you've been in one of these machines. So here you see an iterative reconstruction. Here is the model. On the left is the model that we sort of iteratively reconstruct. This is the target, and here you see the error as it's coming down. So I just want to know how much time I still have.
About 5.
5 minutes. Great. That's fantastic. So we use that to, here you see some results on 5 times knee reconstruction. So the data is from a knee. But, okay, so no, we're reconstructing a knee, but the data could be coming from a brain, right? And then you see how well you transfer between brains and knees. And it's pretty good, actually. So here we are, the knee data, reconstructing a knee, of course, is the best with the most details. But for our model, in fact, given just brain data and reconstructing a knee is not at all bad. It's actually doing quite well, and better than just using a neural net without embedding that information. So then we actually used this, we applied this idea to the Fast MRI challenge, which is a wonderful challenge organized by Facebook and NYU. They give us a lot of pairs of these images, and they said, okay, now at 8 times the number of measurements, 1/8 of the number of measurements, try to reconstruct the best you can, and then we'll measure how well you were doing and compare with others. And so, in fact, in the single coil track, we won that competition with this particular model and some additional ideas that you can, well, were actually presented here at NeurIPS. So then to conclude, so I proposed a model that is related to a VAE. It's not the same, but I think it's also very useful for this community. And it's about deep inverse modeling. So I'm trying to see if the usual way people do inverse modeling can be improved by using deep learning. And I think we have shown that we can do that. So it's a good tool to combine expert knowledge with data-driven methods. We don't need to learn a prior, which you typically have to do when you're doing normal inverse modeling, but it's all embedded in this mapping that you train directly. And it has interesting connections that are yet to be explored to improve a VAE model itself. Thank you very much.
Thanks very much, Max. That was really fabulous. We should send you on more planes, obviously. And I'm going to welcome our next keynote speaker, who's Daphne Koller, going to be in conversation not with Deborah Marks, which is on the program, but with Barbara Engelhardt, and they're going to make their way up to the front now. So give us a second. And if you can, it's great that you're all squashing in, and anybody that needs a seat and doesn't have one, speak up. And if the posters fall down, as I said, we'll do it in the break, and we've got more stickies at the front. And if you want to tweet. If anybody's got stuff on the seats, please remove it so that there's more seats for people to sit on. Slight change to the agenda here is that Daphne is actually going to give us a talk rather than a conversation, which is going to have a last-minute change. So Daphne Koller is going to speak, and she's going to tell us all about the amazing things that are happening at In Vitro, which is amazing, which she will look to you to steal your postdocs and graduate students, no doubt, when she gets half a chance. Take it away, Daphne.
Okay. Good morning, everyone. We're waiting for my slides to load, which I hope will happen momentarily. But we had a little bit of a change in plans because originally these were not supposed to be talks, but then I saw everyone else is giving talks, and I thought and the forum doesn't really lend itself well to a fireside chat. So I'm going to give a talk instead. And I'm going to tell you about a place in which one can, and the reason I thought it might be an appropriate talk to give to this audience is because, as you'll see, we're learning representations that really bridge the different levels of granularity, if you will, that this workshop is all about, where we're thinking about everything that goes from the DNA through the cellular level all the way to the human clinical level. And I think that combination and thinking about representation that really span that gamut is part of what we all need to be doing. And so I'm going to tell you a little bit about how we think about that, and specifically in the context of the very important application of discovering new drugs. So in some ways, one can think of drug discovery from two different perspectives. There's the glass half full and the glass half empty. The glass half full is that in the last 50 years or so, we've made significant dents in a wide variety of diseases that had been thought to be intractable, whether it's cancer, autoimmune diseases, diseases with a strong genetic basis like cystic fibrosis and many others. We're in a world today where things that were once a death sentence or a lifelong of pain are now managed or sometimes even cured. And that's a miracle of modern medicine that I think we shouldn't underestimate. On the other side, the glass half empty perspective is this graph, which has come to be known as Irum's law, which is the exponential. Now, for those of you who've not heard the term Irum is the inverse of Moore. Moore's law is the exponential increase in productivity of pharmaceutical of technology over the last 50 years. This is the exponential decrease in productivity of drug R&D over the same period, where this is a graph on a logarithmic scale, and it shows the number of approved drugs per US $1 billion in spending, and you can see that it's growing exponentially year on year, and the trend is still holding strong. So why is it that a drug now costs $2.5 billion to get to approval? It's not because that single drug is costing that much, it's because the drug discovery process is a 15-year or longer process from idea to approval, where there's many, many forks in the road, where one fork might lead you to success if you're lucky, and 99 will not, and we do not have anything resembling a really good compass that tells us which is the right direction to take. And that's because we don't really understand biology well enough to have strong models of which direction is more correct than others. So if you think of this as a failure of prediction, it immediately lends itself to the question of how can we use machine learning to make better predictions about which fork in the road is going to be the one that is most relevant. So this is where I think we're at the beginning of what is a tremendous opportunity to apply machine learning to these types of problems, this one as well as others. These are two revolutions that have been proceeding largely in parallel. On the one side, the one that all of us in this room are familiar with, which is the advances in machine learning, and I'm not going to belabor how we can do today at beyond human level performance tasks that up until a decade ago would have thought to be, maybe 30 or 50 years from now. The other side, which some of you might be familiar with, is the parallel revolution that's happened on the biological side, and I think Aviv touched on that in her keynote, where we have a set of tools today that each of which has been transformative in its own right, and when put together, provide us with an incredible revolution in terms of data production. These include the ability to take cells from each and every one of us, skin cells or blood cells, convert them into stem cell status, what are called induced pluripotent stem cells, and then differentiate them into neurons with each of our individual genetics, or cardiomyocytes or hepatocytes, that represent the way in which our individual genetics might give rise to disease at the cellular level. We have the ability, using CRISPR, to perturb those cells further, to ask ourselves, if we intervene in the cell and make it more or less prone to disease, how does that affect the cellular phenotype? And then we have the ability to measure phenotypes in unprecedented ways and scales, using things like imaging, like single-cell RNA sequencing, and more types of measurements that are coming online. And what's really important from a machine learning perspective is that now we can do this in a way that is no longer a bespoke biological experiment that takes 4 or 5 years and produces a small amount of data, but we can do this at industrial scale using automation and microfluidics. So those two together, I think, provide the perfect storm of being able to do biology in a very different way. So what we're looking for is to leverage those two threads together in thinking about how to make predictions about this multiple forks in the road that one sees in drug discovery. And we're looking for places where if you had a compass at a particular type of fork, it would actually be transformative. Where machine learning, if you give it the right data, can build a good predictive model. It's not true for everything. It's not the silver bullet. And importantly, because found data is really not enough in a lot of cases, data that was generated for a different purpose and is not fit for what we want to do, we need to generate data that actually helps us construct the machine learning models that we need. And so in what cases can we produce the right data at high quality and at scale? And I think one of the important points, if you were to take away one thing from this entire talk, is the thing at the bottom, which is to do this right, you can't do this by having the two communities working separately in their own little silos and having scientists generate data and throwing it over the wall to machine learning people. This needs to be a truly collaborative effort where the two groups work as a single unit in order not only only to solve the problems, but even ask the question, which problems is it that we can solve? And what we find happening in those few places where this type of community has actually been generated is that we not only answer questions in a different way, we ask a different set of questions because of the interplay between the two different perspectives that come into this. And I think this is a really important thing for organizations as they think about how to move into this new world. So to take a very concrete example of what it is that, that we're working on, there's a number of different things, but I've chosen to focus on this. And this is this is at the core, if you will, of drug discovery, which is answering the question, what will an intervention do when we administer it to a person? And I think that's where most drugs fail today. They fail because we are unable to predict the effect of the drug when it goes into clinical trials. The way in which this is mostly done today is by using mice. Mice are somewhat scalable, relatively inexpensive. You could make interventions in a mouse. You can look at the phenotype in the mouse, and then you have a leap of faith that what you see in a mouse will somehow translate to what you will see in a human. And while that is true for a certain category of interventions, a certain subset of diseases, by and large, this hypothesis has turned out to be mostly false. And many of the, especially for the diseases that occur today, the findings that we see in mice just don't translate well into human disease. And that's because mice don't get Alzheimer's disease, they don't get autism, they don't even get atherosclerosis. So all of these diseases are artificially manufactured in an artificial organism, and then somehow we hope that this will translate over into a human disease. So how do we use humans as a model for humans? Now, that's kind of hard because you don't get to generate a lot of data in humans. We don't get to do experiments in humans, rightly so. So how do we do this? Well, it turns out that we are aided here by Mother Nature because each of us is an experiment of Mother Nature. In each one of us, Mother Nature has perturbed thousands of different genes and has enabled us to see the connection potentially between genotype and phenotype. So when you think about experiments of Mother Nature as driving our discovery process, you actually end up getting a fairly significant benefit. So this is an analysis that's been replicated at this point multiple times that looks at the odds ratio of approval between drugs that have support in human genetics and drugs that don't, and the ones that do are about twice as likely to be approved if you look at the end-to-end process. So that sounds really promising and great. And in fact, we have a tremendous opportunity here, and this is one of the data modalities that we're really excited about because this is one that is growing as Moore's law rather than Irum's law. So this is a graph, again, on a logarithmic scale of the amount of human genomes that have been sequenced since the first human genome in 2001. And not only is this growing exponentially, it's growing twice as fast as Moore's law, so that's pretty striking. If you believe this historical trend, then the number of human genomes sequenced that we will have in 2025 is between 100 million and 2 billion, and that's a lot of data. And some of the work that's been done from the Broad and elsewhere really demonstrates that even just on their own, human genomes provide a lot of input on which genes matter to human disease and which ones don't. That being said, you get even more insight when you juxtapose genetics with phenotype. And this is where, we're really looking at the tie-in between the lowest level, if you will, or the most basic level of human biology and the most significant sort of organismal phenotypes. And this is places where there hasn't been quite as much work as there has been on the genetics, but it's a growing resource. UK Biobank is one of the most significant and well-advanced of these resources that measure genotype and phenotype for the same group of individuals. But there's others here in the United States. There's All of Us that's starting to come online, and there's other biobanks and healthcare systems in other countries where we actually have a tremendous number of phenotypes, again, at multiple levels of granularity in many cases, aligned with human genetics. So this is great, and in fact, now it allows us the opportunity to think about that linkage between the genetics on the one hand and the phenotypes on the other. And because the genetics comes first, by and large, and there's exceptions and caveats, and we can talk about them later, that to this rule, but by and large, this suggests causality. And so that is what has given rise to this other amazing graph, which is the number of associations that have been uncovered in the last decade since the process of genome-wide association aligning phenotype to genotype really started to take off, for multiple diseases as well as non-disease phenotypes. And so you now have this remarkable graph that shows that for many diseases, we're starting to uncover the genetic architecture of disease. And so you could think, well, okay, now we're done. We have, hundreds of gene variants that have been associated with Alzheimer's disease or Crohn's disease, and that's the starting point for a drug discovery effort. Except that it's much harder than that. Because when you think about each of those, and there are indeed, for most complex diseases, hundreds, if not thousands of variants that have some effect on the disease, most of them do not sit within gene coding regions. 90% actually sit in intergenic regions. Most of them we do not understand the function of, and most of them have very small effect sizes. By that I mean something on the order of an odds ratio of 1.03. So you ask yourself, okay, which of those is now a viable drug target, recognizing that it's going to take me years and hundreds of millions of dollars to know whether I guessed correctly? And when you think about that, it's hard to maintain to gain enough courage of conviction to decide which of those forks in the road to follow. And that's where we could really use a compass. So this is, and in fact, when you look at the data, that in fact supports that problem, because if you break down the graph that I showed a couple slides ago, and you divide the targets that have human genetic support to those that have support from Mendelian genetics, where it's really one trait, one gene, and you know what to go after, versus those that come from GWAS, where we really have all this multitude of weak hypotheses, you can see that the odds ratio for Mendelian traits is closer to 3, whereas the odd ratio for targets that come out of GWAS is closer to 1.5, so a lot less compelling. Still signals there, but it's harder to tease apart. So, so how do we get beyond this? So what we'd like to do is to really bring in that other revolution that I mentioned earlier, where instead of just going directly from genotype to human clinical phenotype, you really try and bridge across those layers and have something that looks at cellular biology as an intermediary. And here, what you're measuring is a lot closer to the genetics than the human clinical outcome, where you have all the environmental factors that confound things, the infinite ability of a human to sometimes buffer disease and not have it manifest. And so there's a lot of different pieces that make the genotype to clinical phenotype hard to read, but the genotype to cell biology being a lot easier to read out. And so how do we use that? So first of all, let me give an example of what I mean. This is one of many examples that I could have given, but it's a really elegant one and I think really visually compelling. This is a region in chromosome 16. It's called 16p11.2. It's, for whatever reason, subject to a fairly frequent number of copy number alterations in humans. And when it's deleted, it has a 75% penetrance for autism, and when it's duplicated, it has a 40% penetrance for schizophrenia. Now, the region has 25 genes, sundry enhancers. No one really knows which component in that region, or maybe more than one, is responsible for one or both of these phenotypes. And that's where the field lay up until a couple years ago, when a group at UC San Francisco, with support from the Simons Foundation, took 3 IPS lines, induced pluripotent stem cell lines, from the deletion patients, 3 from the duplication patients, and some number of healthy controls, differentiated them into neurons, and came up with the following cellular phenotypes under the microscope, where even without any fancy processing, you can see that there is a significant phenotypic dose response for the deletion, the wild type, and the duplication, where there is a significant excess of synaptic arborization and axon length in the deletion relative to the wild type, and a significant depletion in the duplication. So this, by the way, is consistent with what you see at the human clinical level, which you can now go and validate by looking at human postmortem brain samples, and you can see a similar phenotype there as well. So, and you can go and by fairly laborious manual process, having recognized that phenotype, go and measure specifically things like dendrite length and soma area and convince yourself that this is statistically significant. And this is great because it helps us understand the pathophysiology of the disease at the cellular level, but it doesn't give us more clarity on how to target the disease. But imagine if we could do this not for 3 patients and 3 controls, but if we could do this at scale, convince ourselves that this is really a robust, reproducible phenotype, and then use this as a screening system for interventions that might revert the cellular phenotype from the abnormal to the normal state. That would give us a certain level of confidence that we were actually going after a target that might also produce a meaningful clinical outcome in the human. So this is where the genetics in some ways ties the cellular phenotype to the human phenotype. And you're using the genetics to create that linkage. So in order to do this, you need to do this in a scalable way. And so what we've actually built is, I need to shoot, sorry. I wish I could shut the music off, but it doesn't let me do that. We've built a data factory that allows you to create massive amounts of biological data at this level. So you take the cells out of the freezer. We have over 100 IPS lines. You put them in the cell management system that allows them to be differentiated completely hands-free. They grow. I wish they grew this quickly, but they don't. But once they grow, again, there's 200 lines. You can put them in this wonderful device called the Echo, which does all of the manipulation of cells and reagents using acoustic liquid handling so that a pipette never touches the medium. So you can do an entire longitudinal experiment in one well and collect longitudinal phenotypes. And then you can take the resulting cells and put them in whatever assay system you have, whether it's a microscope or a sequencer, and basically collect terabytes of data in a single experiment. And we can basically at this point collect about 20 terabytes of images in a single run, which is about a week's worth, and get images that look like this. So it allows you to really do biology in an industrialized way that produces massive amounts of data that look like this. And I love this image because it's nature's art. And I love looking at these images because they're just really gorgeous. So, and in order to now, you have literally terabytes of this, and you can't obviously look at them all by eye, no matter how much you'd like. And so now this is where machine learning comes in. And I think for this audience, especially given the talks that have just taken place, I'm not going to belabor the notion of deep learning and how by doing end-to-end training that starts with the raw data, like the raw pixels or the raw sequence reads, you can go and label them using an automatically constructed feature hierarchy. The only thing that I'd like to highlight from this slide, because it plays into everything that we'll do later, is that in parallel with making good predictions, this also gives you a meaningful representation of the data that is very different from the one that you originally started. So if your original images, say, are 1,000 by 1,000, by the way, microscopy images are 20,000 by 80,000, which makes it an interesting challenge for machine learning. So if you take the images that are 1,000 by 1,000, then they sit in a million-dimensional space. But at the end layer before the classification, you usually have something on the order of 100 numbers, and so you are basically embedding the million-dimensional space into a 100-dimensional space. And so from a mathematical perspective, what it does is it creates a manifold in which the images are actually sitting in this case, a 100-dimensional manifold in a million-dimensional space. And that manifold creates a new distance function that is in many ways capturing something that is relevant to the prediction tasks that you're hoping to solve. So for instance, because the model is penalized unless it labels the two trucks as trucks. It's going to be forced to put these two truck images next to each other in this representation, even though from a purely visual perspective, they are as different to each other as any two random images. The colors are different, the poses are different, there's no similarity at the pixel level. It's only when you look at this representation that they sit close to each other. And because the features are shared across the entire hierarchy, you're also going to have a representation that puts the cars and the tractors, even though their label is different, close to the trucks because they share wheels and elements of the body and so on and so forth. And so again, you've learned the notion of a semantic representation that didn't necessarily jump directly from the data a priori. Whereas other things that don't have that same similarity are going to sit in a very different part of the manifold. So you've learned a semantically meaningful distance function that is really important. So how does that translate to what we care to do here? Here, imagine that we have now images not of cars and trucks and cats and dogs, but rather of cells, like the cellular images that I showed you a few moments ago, and that you're now looking to label them in a way that aligns with human clinical outcomes, because that's actually what we care to do. So first of all, unfortunately, we rarely have iPS cells from enough patients to do this in itself, and so what we can also do is further expand the space of cells by enhancing them using various modifications, using CRISPR, for instance, by introducing a 16p11.2 deletion into a normal cell so that you have an isogenic cell line with and without the deletion, and look at the comparison between them so you can expand the space of cells that you have to have a much broader spectrum, and that then with the appropriate machine learning algorithm, what it does is it creates that low-dimensional manifold in the high-dimensional image space and puts next to each other cells that are coherent in terms of their cellular phenotypes. And what that does is, among other things, identifies clusters in the manifold in kind of a similar way to what we saw for the cats and dogs and trucks. And that's really important because one of the real challenges in drug discovery is that we have been really treating we've been treating multiple diseases with one drug, because Alzheimer's is almost certainly not one disease, and autism is called a spectrum for a reason, and yet we have been trying to look for the one drug that will cure all of these. By contrast, if you look at what has happened in the world of precision oncology, the progress that has been made has come largely by the fact that we no longer think of breast cancer as one disease. We now know that a HER2 positive patient is very different from a BRCA1 patient, and you give them very different treatments. And people who did friends of mine who were Genentech at the time, that Herceptin, which is the drug that targets HER2 positive breast cancer, told me that if you had given Herceptin to an all-comers breast cancer population, you would have needed a clinical trial of about 10,000 women in order to see statistical significance. We don't do those clinical trials, and that's why it's hard to see statistical significance when you treat multiple diseases with one drug. So in the cellular manifold, where you get a lot closer to the biology of the disease, you can see that. And in fact, the clusters in oncology came up by looking at molecular data from enough patients, which you can do because biopsies are common in cancer. They're not nearly as common in multiple other diseases because people don't do brain biopsies for good reason. And so we don't really have the ability to collect enough cellular data on enough patients, but here we can do that. So, having identified those patient clusters, you can now look for interventions because we have, importantly, an intervenable system where we could actually look at the actual effect size of an intervention, at least at the cellular level, and thereby address the question that we had when we talked about genetics at the beginning, which is you have a whole bunch of a lot of variants where at the clinical level, their effect sizes are small. We don't know which ones are going to have a meaningful effect. Here we could actually measure the effect. So this is sort of the high-level picture. This is just to demonstrate that this is starting to work. So this is not all hypothetical. So this is basically looking at how to classify cells using their phenotypes. This is very baby examples right now, but we're starting to see meaningful machine learning in action on these cellular phenotypes. And then this is looking at HepG2 cells, which are cancer cells. I'll get to iPSCs in a minute. Looking at them under chemical perturbations, and you can see that there is significant, you're basically saturating performance. I mean, the computer can see these phenotypes very clearly, even by the way, for doses where a trained cell biologist can't see them. So machines can see things that a computer can, that a person cannot. The first experiment here took about 3 months because it turns out that building the infrastructure on both the lab side and the machine learning side was actually really hard because we're pushing the boundary on scale and quality more than has been done before. But so 3 months work got us to this. Next experiment was getting us a lot closer to the genetics. So now we're not looking for chemical perturbations, which are actually relatively strong, but looking for genetic perturbations and asking, so you perturb the genes using CRISPR-I. This is actually a relatively faithful or relevant, if you will, perturbation to what we see in human genetics because it only changes the phenotype, it only changes the expression of the targeted gene by about 20 to 40%. So it's not that dissimilar to what you would see in a cis variant that is disease causing. And here you see that, the quality of the prediction is not quite as good as you have for the chemical perturbations, but it is still pretty good, and we're only really starting to push the machine learning on this. And what's important is that because we have this closed loop system within the lab where we get to do the experiment, do the machine learning, and iterate quickly, this now took 2 days. And then finally, to show that this doesn't only work for cancer cells, I promised you iPS cells. So these are neuronal cells that were differentiated from iPS cells. This is still only a chemical as opposed to a genetic experiment. But again, if you show this to a biologist, they often can't tell the difference between the different drugs. And you see a pretty good clustering in the manifold that we have for the labels here. And again, the quality of the predictions is getting quite high. There's still room to grow, but you can clearly see evidence of mechanism of action from these images. So maybe just to sort of summarize, I think coming back to maybe the main philosophical takeaways from this talk. First of all, in order to do this type of work, I think it's absolutely critical that you have an incredible collaboration between people with very different types of expertise that normally do not talk to each other and normally do not have a common language. And you need to put them together, and you need to make sure that they have that they're thinking together about the problems, not at the very end when the data has already been produced and you ask someone to analyze it and you explain what the data mean, but rather from the very beginning of which questions should we even go after, which experiment should we do, how do we build the assay, how do we QC the assay, how do we build the model, how do we interpret the results, and how do we feed that back to the next round of experiments, has to be done as a single unified collaborative effort. It needs to be done in a closed loop with significant iteration and the ability to sort of do rapid experimentation, and that's where automation can really make a difference, to the ability to not only generate data at scale, but also generate data quickly. Because one of the things that's transformed machine learning, for those of us like myself or old timers who lived through this, used to be that it took you 9 months to write machine learning code in C++ and 6 months to run it on your local cluster, and you hoped that there was no memory leak and the thing actually ran. And then if it did, that was the only time you ever did that. And now we can do something in TensorFlow in a week and run it in the cloud in 6 hours, and you have the ability to look at your results and iterate and make them better. We can do that with biology now, not in 6 hours, but experiments that used to take years can now take weeks or maybe even days, and that is going to be equally transformative to our ability to make the results better as it was to machine learning, but it has to be done in a way that allows that rapid experimentation. The last piece that I'd like to talk about is maybe just jumping up a level to the very highest level of granularity, which is just to think philosophically for a moment about epochs of science. If you look back at history, there's been periods where a particular discipline has made a tremendous amount of progress in a very short amount of time. And that's because there was a particular invention or perspective that allowed this rapid progress to be made. In the 1870s, that was chemistry, with the understanding of the periodic table and that you couldn't turn lead into gold. In the early 1900s, it was physics with the understanding connection between matter and energy and space and time that still drives a lot of physics understanding even today. 1950s, it was obviously digital computing with the ability to do in silicon chips calculations that only a human, or sometimes not even a human, had been able to do. And then in the 1990s, there was an interesting bifurcation. One field that made a tremendous amount of progress is the field of data, which is related to but different from computing in the sense that it also involves statistics and optimization and various other pieces. And so I think of it as a separate field, even if it grew out of computing. And the other is the field of what you might think of as quantitative biology, which moved biology from purely observational science where you taxonomize animals and such and plants into something that was much more measurable and quantifiable in a completely new way. And that emerged from sequencing and microarrays and super resolution microscopy, and it was a complete transformation in our ability to understand biology. But these two fields largely evolved separately as separate entities. And the field that I think is coming in the 2020s, and I think that is going to be the discipline of the next epoch, if you will, is what you might call digital biology. It's the ability to measure biology in whole new ways at unprecedented fidelity and scale, to interpret what we measure using the new techniques that are emerging from machine learning and data science, but not just any old techniques, things that are specifically going to be driven by the needs of this new field and this new data modality, and then taking those insights back and translating them into editing biology, manipulating biology to do things that it wasn't meant to do before. And there's going to be repercussions for this across multiple disciplines, in environmental science, in material design, in agriculture, and in human health. And I think it's going to present remarkable opportunities to push forward the technology on both those sides by virtue of having those two communities work together as a single whole. And so I'm hoping this workshop helps catalyze some of that collaboration and help us make progress in this space. So thank you very much.
Okay, thanks so much, Daphne. That was fantastic, so exciting. Somebody actually walking the talk as well, not just saying the vision, but actually doing it. Amazing. As I said, we're going to not have questions now because we've got such a packed agenda. Take a 15-minute break. We'll come back at just before 1:45. And take the time to put your posters back up, the stickies at the front. Talk to people. Grab the speakers.